So I'm going to just start with the introduction. And we let people join in as they go along and watch them later, the recording. So I am very pleased to introduce Dr. Pandora Pound, who is the Research Director at Safer Medicine Trust and is going to be talking to us today about the rat trap. I'm really pleased to welcome her and excited to hear this presentation. So I shall hand over to you.
Thank you, Eva, and thank you very much for the invitation to talk today. I was really interested to hear about the Humanimal Trust because I hadn't really heard about it before. I had heard about One Medicine, but I was thinking, oh, that's quite different from what we do, and I hadn't really gone back to it. But the more I've thought about it, I think there's actually quite a lot of similarity in our aims. So I'm really pleased to be giving this talk. So yes, I'm from the Safer Medicines Trust, which is a UK-based patient safety charity, and we really aim to encourage the use of human-based methodologies in the development and testing of medicines, because we think that will make medicines safer and more effective. So I was intrigued to learn about the Humanimal Trust's emphasis on what humans and animals have in common, because in fact, at Safer Medicines Trust, we emphasise the differences between humans and animals, because we argue that this makes it difficult to use animals as models for humans. Yet, of course, we both, I think, want to move away from experimental animal models. So I'm interested in seeing, what we have in common. And I'm going to just talk today about Rat Trap, which was a book I wrote, which was published in 3 months ago now, at the end of August. And I'll tell you a bit about why I wrote the book and then what it's about, and thereby hope to show that although we're coming from opposite directions in some way, we've got very similar aims, I think, so our positions aren't as contradictory as I might have thought initially. So my interest in this area began in the 1990s when I was working at St Thomas's Hospital Medical School. I was working as a postdoc doing stroke research. My background's in the social sciences, so I was working with a team of social scientists on stroke, and my office was actually quite close to the animal lab there, and occasionally, or rarely really, I saw animals being brought in or out through these service tunnels that used to run underneath the medical school, and they used to pop up here and there, and one of the places it popped up was in our department, so I would occasionally see people going in and out with trolleys with sort of partially covered cages and things. A couple of years later, the site was abandoned and we moved to another site within London, but many years later, some urban explorers went in and photographed the site, and I was able to see, years later, what had actually been behind these closed doors. So you can see here this abandoned laboratory. And I became curious about animal research. I noticed when I tried to talk to my colleagues at the university, at the medical school, that it was a very taboo subject. People really didn't want to talk about it. And I then became more interested really in the extent to which it benefits humans, because that's the way it's justified. And when I went to look for the evidence about this, all I could find were these very generalized statements, such as animal experimentation is a valuable research method, which has proved itself over time. That was from the House of Lords report of the Select Committee in 2002. And in the same year, this statement of the Royal Society, virtually every medical achievement of the last century has depended directly or indirectly on research with animals. The need for animal research is self-evident. So I didn't think it was self-evident, and when I went looking for the evidence, as I say, all I could find were sort of isolated examples. I didn't find any robust systematic evidence to support these statements. So I looked into it a bit further, and my investigations resulted in a paper that I wrote with colleagues in 2004, professors of epidemiology. And this paper was published in the British Medical Journal, and it was really asking where was the evidence that animal research benefits humans, we said we found very little robust systematic evidence, and we felt that it really wasn't good enough just to make assertions about its benefit. And so we were suggesting that one way to look at the evidence would be to use systematic review methodology. So over the intervening years, animal research did come under a lot of scrutiny in a way that had never been before. So there were systematic reviews conducted which looked at the scientific rigor of animal studies, which looked at the translation, the predictive ability, its use in drug safety. And after probably, 10 years or so, some conclusions were possible to draw. And it was certainly seen to be the case that animal studies were not conducted according to accepted scientific standards. And because of this, much research was unusable. And there were grave doubts about clinical translation. So I was actually really shocked about these findings, because I thought in a morally contested field such as animal research, surely scientists would take care to do their research properly, make each animal's life count, and produce useful findings. And I wanted to make this evidence available, both about the limitations of animal research and the lack of clinical translation. Because although it was a sort of open secret in the scientific world, I think much of the general public was still thinking that it was a necessary evil. And I wanted the book to focus on the science rather than the ethics, not because I thought ethics was unimportant, but because I thought the ethics of animal research had been well covered. And I wanted to look at the impact of animal research on humans rather than animals, again because I thought the impact on animals had been well covered already. I wanted this book to be affordable and accessible because I wanted ordinary people to read it. And the aim really was to change people's hearts and minds and hasten a transition away from animal research and to the human-based researches that we think would be more effective. So Rat Trap, that's what it came to be called, has 4 parts. I talk about the origins of animal research, scientific limitations and lack of translation, the human-based approaches that we feel could replace animal research, and the obstacles to transitioning away from animal research. And the book is based on evidence, the best evidence I can find. But to make it a little bit more engaging, I also interviewed experts and drew on some of my personal experience. And I'm now just going to give you a quick sort of whistle-stop tour through the book, starting with these historical chapters. So I talk here about the enthusiasm about the experimental method, which was emerging in the 19th century, and particularly in France, because the experimental method was associated with proper science, so like physics and chemistry. And an early exponent of this method was Claude Bernard, who was a physiologist and a doctor. In 1865, he wrote this book, An Introduction to the Study of Experimental Medicine, in which he argued that studying animals under controlled laboratory conditions and then extrapolating the results to humans was a more scientific way of going about studying humans than actually observing them directly. And his aim was to place physiology on a firm scientific footing and to make it into a hard science. So he believed that biomedical science should be conducted in the laboratory, and he rejected evolutionary theory, which was just emerging at the time. Darwin's Origin of the Species was published really around the same time as Bernard's book. And Bernard believed that all animals shared what he called the same vital units. And so he believed that species differences were quantitative rather than qualitative. So they were differences of degree only, which could then be accommodated using scaling techniques. And academic animal research, I think, is still very much under Bernard's influence all these years later. So animal experiments served to create distance from the patient's bedside, making biomedicine more scientific. It came to be seen as a robust scientific approach because it drew on the use of statistics, and there was an ability to control variables and so on. And there was therefore a consequent reluctance to criticize the practice among other scientists. But I ask in the book who benefited from these animal experiments. Certainly, historians agree that they were very successful at raising the status of the professions of physiology and medicine. But even towards the end of his life, Bernard himself was unable to give any examples of how his approach had actually benefited humans, or benefited or progressed medicine when he was asked to do so. So in the next historical chapter, I then look at how animal experiments became locked in over the 20th century. And here I look at the rise of animal breeding programs, because scientists wanted sort of so-called standardized healthy animals to use in experiments. And so these breeding industries grew up to supply scientists. They also look at the influence of the world wars, because there was a great demand for animal research because of the world wars, and demand for experiments on chemical warfare and radiation and so on. The rise of the pharmaceutical industry, and also national institutes of medical research like this one that used to be at Mill Hill, north of London. The UK's Medicine Act in 1968, until we've got the situation now where animals are now embedded in our institutions, our educational institutions, our medical institutions. And so we've gone from a situation in 1881 where there were 250 experiments recorded in the statistics. That was the first time the government recorded those statistics, to 95,000 in 1910, and then peaking at 5.6 million in 1971. And so the way animals are used mostly in biomedical research now is with animal models, experimental animal models. And this means that the animal is manipulated in various ways to try and make it mimic human disease. So, for example, in stroke research, animals will have a stroke artificially induced, typically using surgery to block the middle cerebral artery. And then an experimental drug is tested on those animals and the outcomes are assessed. So I then go on in the book to look at animal research methodology and the outcomes. So this is really looking at the limitations of animal research. And here I'm looking at the studies themselves and looking at internal validity. So that's experimental design, attention to bias and so on, but also external validity. So the way in which these studies are supposed to be generalized to diverse human populations. And the issues with external validity, I think there are 4 different issues when it comes to generalizing from animal models to humans. So there are problems with the models themselves, and I will go on to talk about this in a bit more detail. There's problems generalizing from homogeneous animal samples to diverse human populations, from the laboratory context to the clinical context, and then of course from animal species to human species. So just to look at internal validity in a bit more detail. So systematic review evidence showed that a few animal studies were taking steps, or do take steps, to minimize the risk of bias. So only about a third of studies were found to be actually randomly allocating animals to treatment or control groups. And between 21 and 35% were actually conducting their outcome assessments blinded. And less than 1% of animal studies reported a sample size calculation. There was also widespread failure to control for confounding variables such as the animal's strain, sex, age, and other variables such as the animal's bedding, housing, and also how the medication was administered, and so on. There are also issues with reporting the animal studies. So the loss of animals from studies is poorly reported, and that of course affects the findings. Analyses with the best results have been found to be selected for publication, and of course there's the issue of non-publication of negative studies. Now the trouble with this, all these biases make animal studies appear much more positive than they actually are. So other issues with external validity, so going back to the 4 categories I talked about, so the models themselves. So in humans, most human diseases evolve over time, and most animal diseases too. So, for example, humans get stroke due to risk factors that accumulate over their lifetime. So, for example, drinking, smoking, high blood pressure are risk factors for stroke. But animal models generally mimic a very advanced stage of the human disease, often the final stage. So going back to stroke, as I said, this typically involves surgery to block the middle cerebral artery, but that's the very end stage of a process that might have taken years to develop in a human. And the samples, laboratory animals, typically young and healthy, whereas most human diseases manifest in older age and often alongside comorbidity and polypharmacy. And laboratory animal populations are highly standardized. They're genetically very similar, but of course human populations are diverse, with all sorts of variation in disease susceptibility, immune response, and reactions to drugs and so on. And another issue in the category of external validity is context, as I mentioned. So trying to generalize from the clinical to from the laboratory to clinical context often raises problems. So Terilizad was a neuroprotective drug. It was developed for use with stroke patients, and when it was trialed in animals, it was found to be successful if given within 10 minutes of the stroke. But of course, it's impossible for humans to actually realize they've had a stroke, access acute stroke expertise, have a CT scan to confirm that, and then get a drug administered within 10 minutes. In clinical trials, humans were given Terilizad within a more realistic 5 hours, and of course the trials were unsuccessful. And finally, species differences. So animal models are based on a hypothesis of homology, which goes something like, because of biological similarities between humans and mice, for example, what's found in mice will apply to humans. But of course, while species may be biologically similar, they're also likely to be significantly different biologically because they've adapted to different ecological niches. And you can see this here with the finches. They look similar, but they've adapted to different ways of eating, and so they have different beaks. And an evolutionary biologist, Robert Perlman, wrote a very interesting paper in 2016 about species differences. And he argued that, of course, mice and humans have genetic, biochemical, and physiological similarities, but of course the lineages that led to modern rodents and primates diverged around 85 million years ago. So mice could, of course, help us understand processes that arose early in evolution, but now we've got really different life histories. Our diets are very different. We're exposed to different toxins, pathogens. We've got different microbiomes, immune systems, and so on. So he argues that mice aren't useful for understanding chronic non-communicable diseases, those diseases that we suffer from, because the causes of these are enmeshed in our unique evolved life histories. So the theory of evolution explains why animal research doesn't translate to humans consistently. Sometimes it does and sometimes it doesn't. It can be a bit hit and miss, but we know why. And in complex living systems, even minor differences can result in very significant differences in biological processes and outcomes. And that's, of course, really important when it comes to developing drugs. Non-human primates are often cited as having great genetic similarity with humans. And it was for this reason precisely that experimental drug TGN-1412 was tested in non-human primates. It also underwent tests in a range of other species, and no issues were raised from the animal tests. But as you probably all remember, within minutes of receiving this drug, 6 healthy young men went into multiple organ failure, and it was a drug trial that was really infamous. It was known as the Elephant Man drug trial. It took place at Northwick Park. A horrible incident. And afterwards, an investigation showed that the reactions were the result of an unpredicted biological action. A few years later, something similar happened in France, when, again, 6 healthy male volunteers were given a drug BIA 102474. This was in France. And after taking it at a certain dose, one man died and 4 others were hospitalized, 2 of whom were left with residual neurological impairments. This drug had also been tested in mice, rats, dogs, and monkeys, and they'd raised no concerns. But tests later conducted on human cells found that the drug disrupted metabolism of human nerve cells, and this, of course, wasn't possible to identify using animal tests. Even approved drugs can cause adverse reactions and deaths when used in the wider population. So triglycerone was approved in the late 90s in the US for diabetes, but was withdrawn 3 years later after reports of deaths and severe liver failure. Again, the animal studies hadn't identified any cause for concern, but tests in human cells and tissues conducted after the disaster showed strong indications that the drug would adversely affect the liver. And in case you think I'm just picking the bad cases from a sort of sea of successes, there was a really good study which was done on 2,000 drugs, comparing animal and human toxicity data. And it found that the presence of toxicity in animals does correlate, although not very reliably, with the presence of toxicity in humans. However, the absence of toxicity in animal tests is unable to reliably predict an absence of toxicity in humans. In other words, if a drug appears safe in animals, it can then go on to be toxic in humans, as we've seen in all these examples. So animal tests are failing to safeguard humans. But this lack of predictive ability, it also leads to unmet need in terms of treatments and cures for diseases. So there are hundreds of common human diseases for which we have no cures and treatments. I mean, we all know people who suffer from illnesses for which there aren't any treatments. And in fact, only 8% of experimental drugs make it from phase 1, first in humans, right through to approval, as you can see here. And animal studies are widely acknowledged to play a key role in this low success rate due to their poor ability to predict human safety and efficacy. And you can see that it's, if you look down at the bottom, in oncology, 5.3% success rate, cardiovascular, 4.8. I mean, this is terrible. Neurology, 5.9. And if you look within these categories, Alzheimer's, for example, has a 0.4% success rate in terms of developing beneficial treatments for humans. Sepsis, leading cause of death in hospitals, no treatments at all, despite decades of animal research. Same with traumatic brain injury, stroke, 0.1%. So we're not doing well using animal studies. Just to look at stroke in a little bit more detail. We've had animal models of stroke since the Second World War, but there are still few pharmaceutical options for patients with stroke. Numerous drugs have been found to be successful in animals. For example, over 1,000 potential neuroprotective drugs were tested in animals, and although many were found to be beneficial for animals, not one benefited humans. And in fact, some of them increased mortality in humans. There's only one drug for acute stroke, and that's a clotbuster called alteplase. This went straight into clinical trials following success with heart attack, so it wasn't even a result of animal models of stroke. But even this one drug can only be taken by about 10 to 12% of stroke patients. And of these, half will remain dependent or die, so even this can't really be counted as a success. So I then go on to talk about, in the third part of the book, human biology-based approaches, which we feel is the way to go forward. And here I look at in vitro approaches such as organoids, organs on chips, in silico and AI, and early detection approaches. For example, the NHS is currently piloting a blood test in thousands of people who don't have any symptoms of cancer, and the aim is to use a single blood test to detect, I think it's over 50 cancers at their very earliest stages. And this was developed on the basis of human-based research conducted by Professor Azra Raza, and that would be a massive game changer if that turns out to be successful. I also talk about more tried and tested approaches such as scanning, prevention, epidemiology, post-mortem studies, clinical research, microdosing, and real-world data. And I argue that we have such a huge range of human-based approaches that we can use either singly or in combination. That it doesn't make sense to use animal models. I'll just look at a couple of these in a bit more detail. So this is the organ on a chip, and this is taken from Emulate's website. This is a company in the United States that develops organs on chips, and they've developed, as you can see, brain chip, colon, duodenum, kidney, liver, and lung chip. So the organ on a chip is tiny. It's about the size of a computer memory stick. It contains microscopic channels that can be lined with living human cells taken from an organ. And you can pump blood, air, and nutrients through these channels. And the idea is to closely mimic the dynamic microenvironment that cells are exposed to within human bodies. So a sort of home from home for the cell. And last year, a really great study was conducted which shows the value of this approach. So the scientists took 27 drugs that had been judged safe for human use based on animal study evidence. And you can see here some of the ones, diclofenac, triglyceride, fluoridin, simvastatin, but which had then gone on to cause serious adverse reactions in humans, including liver failure and death. Fluoridin, for example, resulted in clinical trials resulted in the deaths of 5 out of 15 patients, and only 2 others were only saved with liver transplants. So we're talking serious reactions here. So they tested the drugs in 870 liver chips. You can see that there a bit more closely. And the liver chips were able to detect toxicity in almost 7 out of every 8 drugs that were toxic to the human liver, so far outperforming tests in animals. So you can see the potential of this approach. And then there are in silico approaches. So you can feed knowledge, for example, about the characteristics of a drug or drug type into a model, together with information about the biological system into which it will be introduced, and knowledge from trials and other sources about how that drug works. You can sort of feed that into your model, which will then predict how that drug will behave in humans. And there is a model called DILISIM, so DILI is drug-induced liver injury, which has been developed to predict whether new drugs will cause liver injury and to elucidate mechanisms. And it was able to predict that the migraine drugs telcagipant and MK3207 would be toxic to the human liver, which led to their development being terminated despite the fact that animal tests had raised no concerns. But it was also able to predict that ubrogepant, or a related drug, would be relatively safe for the liver. And this went on to be confirmed in human trials, and the drug was then approved by the FDA with no safety warnings, which is quite unusual. So this model was actually able to predict both the presence and absence of toxicity, something that animal studies are unable to do. So in the final part of the book, I look at the obstacles to progress, obstacles to transitioning away from animal studies. And here I look at things like the regulation of animal studies, the regulations covering the production of pharmaceutical products, the focus on refinement rather than replacement in the 3 Rs, the whole project to improve animal research rather than replace it, the ongoing perception of animal studies as the gold standard, and the fact that animal studies are firmly embedded within our universities and institutions, as I mentioned earlier. Together with the fact that there are vested interests, there are huge industries supporting animal research, supplying not only animals, but cages, equipment, food, housing, and so on. So there's a lot of money to be made from it, but not enough money available for these for funding human relevant studies. There are also organisations that exist purely to resist the transition away from animal research, such as Animal Research Tomorrow. But there's also good news, and I talk about this in the book. So, for example, at the end of last year, the FDA voted to, they passed the FDA Modernization Act, which means that scientists developing drugs in the United States can now use any approach that they believe to be scientifically appropriate. So it doesn't ban the use of animals, but it means that scientists are now open. They don't have to use animals. They're now open to use whatever they think is the best way, the best approach. And this has really created some momentum and some optimism about moving forward without using animals. So that's the book. I was really pleased that it's had some really good reviews, which was good to see because it was quite hard to get this book published, but that's another story. So I'm just going to leave it there really with, I thought our approaches appeared contradictory because we focus on differences and the Humanimal Trust focuses on similarity between animals and humans. But of course, we're both concerned with improving medicine. We both have doubts about the utility and the ethics of experimental animal models. And evolutionary theory shows that animals and humans can have similarities, but they also have important differences. So I think there's room for both approaches. So I think our approaches are complementary. So that just leaves me to thank Kathy Archibald, my colleague, without whom I would not have been able to write this book, and all the supporters of our Safer Medicines Trust who've been so generous in keeping us going. And yeah, a little plug there about how to get hold of the book. Thank you.
Thank you very much for that. It was really informative presentation. I've been busy writing loads of notes and things I have to look up. Just in terms of some of the questions, it's really interesting to see some of the things sort of that happen in terms of the types of experiments that go on. And one of the questions I had, what you mentioned quite early on, was do you think in any sort of clinical trials, there has been for quite a few years the sort of demand and the need that every single trial, as part of the transparency drive, has to be actually registered on a public database to disclose before the research even starts as to what's going to be happening? Do you think if there was an equivalent that existed for, obviously, animal experiments, because currently most governments publish sort of annual figures, but still very much cloaked in uncertainty as to what some of the usage is. Do you think if there was an equivalent sort of demand in the public domain that would actually make people perhaps rethink some of the experiments that are actually taking place?
I think there is people who conduct systematic reviews of animal studies are encouraged to register their protocol in advance, but the animal studies themselves, you're right, yes, they're not registered up front in the same way as clinical trials. I think it would make a difference because there'd be transparency about the hypothesis, so that wouldn't be possible to change that halfway through. There would be hopefully transparency about the sample size and so on. However, I don't go down the route of trying to improve animal studies. I just think we've got to replace them.
And I'm not thinking in the context of perhaps improving them, but thinking in the context of if people actually did have to do that perhaps they would be thinking a bit more about if they actually need to do this.
Well, yeah,
definitely. There seems to be quite a lot of that.
I think there is, there are calls for people not to even be able to, propose an animal study until they've conducted a systematic review of all the available evidence, and that would answer your question. No, they would, do I actually need to do this study? But of course, that's not mandated, and I think it's quite a big issue. It's quite a big undertaking to conduct a systematic review. So I imagine that some sort of informal reviews are conducted, and of course they're very subjective, and it's always very easy to say, oh yes, there's a need for this animal study. The trouble is most animal studies are justified because they're of some claim regarding its human benefit, but I mean, the history and the evidence shows that there's so little human benefit that it seems unlikely that this particular one is going to suddenly, break the mold and be successful.
Mm. And it does see, I mean, some of the figures that you quoted are quite shocking, isn't it? That only 8% of actually anything that enters a phase 1 trial, and that would have taken quite a lot of steps already, actually gets to a full approval stage. And of course, when we then talk about the approval stage, and you mentioned some of the medications as well, even in the post-approval, because of course people suffer from lots of comorbidities, and the trials are not really designed to capture that.
No, it's only once they're trialed in the huge sort of population that you find
that there isn't. It seems quite a shocking figure in terms of the translation that we persist in that sort of an approach. One of the other questions I was thinking about, and is sort of also linked to conversations we've had with other people, and I have tried looking, but I haven't seen. Do you think having a sort of an internationally supported database of alternative methods for specific experiments would help to actually access people better ways of conducting some of these experiments? Because currently there doesn't seem to be anything, and quite a few of the scientists we've spoken to is that they say that they're very unsure about what are the validated alternatives. There doesn't seem to be a way to sort of a straightforward way to actually find what these are, and even though they would be really willing to use them, there's just a lot of uncertainty, because from their perspective they get criticized by their colleagues if they try to opt for difference. But if there was actually something a bit more recognized where they could go to, are you aware of anything like that being created?
There are several databases. Unfortunately, they're not very well known about. I'll have to let you know what they are later, because off the top of my head, I'm afraid I can't remember, but there are several in existence. Though, I mean, there are a few issues with that. One is that, I mean, certainly in the UK, there's no requirement by the regulators that, I mean, they're asked, when they submit a proposal, scientists conducting animal research, as you know, are asked to say whether they've tried to do the research using alternatives. But there's no requirement at the moment to actually show that the investigations they conducted, did they look at certain databases? Have they actually tried this alternative method? They can simply just write, there was nothing suitable, So they're not required to actually show the work they've done to find any alternatives. So even if there, are sort of well-known databases, the fact there's nothing actually going to make them use those databases at the moment in the UK anyway. There are people, as you say, who want to use them and who need who may want guidance in how to use them. And there's a fantastic initiative in the Netherlands. I don't know if you've heard of it. It's called the Helpathon. Yeah, and it's when people who are doing animal research and who want to do it differently, want to use non-animal methods, put out a call for help, and people in the area who know about the alternatives will say, yes, I can help, and then they get together for 2 days. There's always a night in between so that you can think things through and socialize in the evening and so on. There's actually dialogue, really focused dialogue over a couple of days, and at the end of it they come up with a plan to do the research without animals. And I think this is just such a fantastic way ahead, because it's dialogue rather than opposition. And I think it would be something that would be really great to use in the UK. There's been one in the UK, but it's a great model. It's a great way forward, I think.
It does certainly sound like something, and I think that's the important bit that you say is about having the dialogue.
Yes.
Because often people get a bit too ingrained in their ways and feel endangered and assaulted when somebody tries to put forward different options. So having the dialogue is really, really important.
And I think in the helpathons they make it very clear that it's a very friendly, safe kind of learning environment, and it's not about scoring points or opposition or anything like that. It's just very collaborative, and there's been so much good feedback about them. They're great.
I'm going to, Andrea has got her hand up. Before I let her ask a question, just one quick sort of, what do you think from your perspective is the biggest really obstacle for people not sort of, not being more open to non-animal alternatives that now exist? And I mean, I know it's sort of, in a way, organoids have been around for 15 years. It's still very early type technology. So, but as more and more is happening with the in silico studies that AI can help to deliver, it seems strange that people are not more open to these alternatives. I wonder if you had any thoughts as to why that is.
I think, well, I think to some extent the AI and organoids and organs on chips and these approaches are being used, but being used in tandem with animal research in many places, certainly within pharmaceutical companies. And it's, yeah, hard to know why, but anyway, maybe it's just like a phase in this transition that takes place. But in terms of in terms of obstacles, it's not so much that the science isn't there. I think it's that the animal research is so locked into our systems, into our regulatory systems, into our institutions, um, that it's like turning around one of those huge tankers. It's just really hard to unpick it and prize it out, because a lot of people's livelihoods depend upon it. Whole university departments depend upon animal research, journals, conferences, PhDs, people's careers. Apart from that, regulation of animal research isn't being properly applied. So every single animal study that's submitted to the UK regulators gets approved. This is something that's supposed to be a last resort. So I could go on. There's a lot of issues with the obstacles, but there are a lot, and we have to work on them all, I think.
No, I mean that is very helpful, and I think in many ways reflect a lot of the discussions that we have with other people. And I think putting forward, and we've been trying to through some of the groups that we part of to actually start perhaps that there should be initiative to actually make it a legal requirement to use alternative methods if they exist, rather than leaving the options of where possible and leaving the wording within the legislation so loose that we would just keep reverting back to what they have always done. Yeah.
Yeah, definitely needs to be tightened up.
Yeah. Angia, do you want to go
Hi. Yeah, so some of what I was going to say is just kind of what Pandora's just covered. So the University of Manchester had an open debate earlier this week on the use of animal research. Yeah.
How did it
go? Yeah, it was very interesting. I was actually, if I'm honest, I was surprised how it's not pro animal research, permissive of animal research. People were, so there was somebody from the RSPCA, there was somebody from Frame, and there was somebody else who I can't remember where she was from, kind of more on the sort of medical side. And all still, even the RSPCA recognise the difficulties in changing the use of animals in research. I'm at the University of Manchester at the minute, and one of the kind of senior lecturers and senior leadership people there was saying that they do animal research even though they don't want to. And some of that is because of the reasons you've said, Pandora, because it's kind of required. They work in neurology, so it's kind of essential for that field. But something else that was brought up that was, and I know this is a big problem in academia, is that actually getting funding to use the alternative methods is incredibly difficult because the funding bodies and the review boards don't have specific knowledge of the alternatives. So when they come through the system, they're actually quite wary and reticent of awarding funding for something that isn't proven or isn't widely published as being proven.
Yeah.
And we kind of I was having a look around afterwards, and I think for the alternative methods, I think funding is something, I think last year was around about 11 million or something. But then for the MRC overall funding budget, it's in the billions. So there's like massive magnitude difference there in what can be funded and how that is funded and taken forward. And I think within the university culture, that's huge. It's not only kind of established academics that this is what we do, this is how we do it, and it's very difficult to get them to change. But actually, where you have early career researchers coming through and wanting to change that, they're hitting a brick wall when they come to apply for funding, because these methods are just not widely enough known and proven. As you said, Pandora, you know, the animal research is the gold standard. And I think it's quite difficult. It is like turning a tanker. It's a really big issue to address, to get that culture change through all of the kind of universities at all the different levels, but then also through into the funders and the funding bodies to kind of see that as a positive.
Yeah, I mean, you've said it all, really. I mean, if I had a magic wand, I would sort of tighten up the regulations, but I would also sort out the funding, because basically, if you've had a lab and you've been doing all your research using animals, and all your publications use animals, and your PhDs use animals, you're not going to be able to just suddenly start using a different methodology unless you're incentivized with funding. And that, you know, scientists follow the money, don't they? They follow the funding. And so if that dried up overnight and they were funded to make that transition and supported in that, that's how it would happen.
Yeah. I think another area as well, I always think would be quite an interesting thing to do, and this relates to the NHS and the sort of systems within the NHS, because I think there'd be great value if they were much more married up and aligned throughout the UK. I think you'd get much rich data and more informative data on conditions that people have and issues that come up with those conditions, and then you could feed that into kind of AI and other systems. So, because quite often, I don't know if you all know how the NHS works, but kind of different parts of the NHS are all separate. So your primary care generally is different to secondary care. It's not linked in. Manchester is different to Kent, is different to London, and they're not kind of aligned and linked into each other. So there will be different, obviously people have strokes throughout the whole of the UK, and there will be different things going on with strokes, but none of that is fed into anywhere where it can be mined to look for things like markers for early detection. Everybody's sort of working in silos in their own area, and I always think that's a really big shortfall of the NHS.
Yeah, I mean, there's so much information, isn't there, that we could be using.
If you kind of even confidentially have access to that patient information, you can kind of pick up on things earlier that can then help and feed into, and then perhaps you would have better indicators of early detection.
Well, I think, I mean, that's also called real world data, isn't it? And I think it's used a lot with, certainly with COVID, and has been used, but it could be used to so much better effect, as you say, definitely.
Yeah, I mean, COVID's a really good example of that they can do it when they want to, I think.
Yeah.
Because that, you know, huge things happened very quickly with COVID. And so the possibility and the potential is there. It's just not being tapped into.
No, all really good points.
I think there is some drive through the NHS Digital to try and utilise the data a bit better, but obviously, like everyone, because of the fact that you mentioned that the systems are so desperate that you can't actually, it's not so easy. You would have thought is one NHS, we should be able to utilise the data better, but actually pretty much every hospital uses a different system, never mind the regional, and it then just becomes so complicated. But there's certainly a big push from NHS Digital to start delivering a wider range sort of digit trials, as they call them, but gathering the data and having access to the data, it causes the massive issues. And I think it's a similar sort of thing that they're trying to access and talk about in veterinary medicine as well, where they would like to take some of the data together, similar sort of approach to see if they can find things. And there are various initiatives around the country, but that being even more complicated because obviously
because it's private.
It's private, so it's there's a lot of potential there, but there's also a lot of technical and legal challenges around pulling some of this together. But it definitely is a great option to look at.
I think if you combine that with the AI that we've got going now, I think that will be very powerful in the future, long term.
Yes. In the future.
Well, AI was very helpful in COVID as well because it was able to sift through like thousands of drugs and drug types to
Mm.
select ones that could be, potentially useful, and it did that in minutes, whereas with animal studies it would have taken, years. But
Yes, definitely. So did you have a question? I noticed you had your hand up.
Yeah, a bit of a sort of a statement and a question, if you like, Pandora, and hopefully not too controversial at all. But just that, going back to your points about, animals, whereas Humanimal Trust very much about looking at that connection between humans and animals, but also connections and collaborations between the different human and animal medical disciplines. And I think one of the reasons there's a couple of reasons we do that. And one is because we want children from a young age to understand sentience. And I think sentience is a word easily brandished around, but I think when you can start introducing that to children from sort of as young as you possibly can, from primary school age, as to actually how similar we are from a behavioural perspective, how we all suffer grief and joy, and we play, we're involved with teamwork, all of those different areas. It really truly helps those children understand what sentience actually means and how similar we are. And then obviously going up the ages and looking at the clinical trials, there are certain diseases, naturally occurring diseases and injuries even, whereas the treatments for can be relatively the same. But by learning from those clinical trials without experimenting, we can actually really help many lives. So I think that's where we're sort of coming from. But what I'd be interested in, I suppose my question for this now is, psychologically, there's been a lot of work done lately on things like PTSD, Alzheimer's, old age, and the effects it can have on both humans and animals, anxiety, depression. And we know animals can suffer those traits as well as humans. And there is much work being done on clinical trials with those areas. And I just wondered what your thoughts were on that.
Well, I think until Eva got in touch with me, I hadn't really thought about it at all. And I was delighted to hear that. I mean, it just makes sense, doesn't it?
Mm.
As long as the ethics are there in terms of the animals being consented by their, companions or whatever we call them.
Their guardians.
Their guardians. Then it seems to make sense to me completely.
Yeah, and I think that's what we're trying to do. We're trying to, obviously we try and fund research, but we will never fund research on experimental animals. That's just not what we do. So what we want to find money for and then fund is either looking at alternatives or on bringing about those collaborations and connections to bring those different medical disciplines together to actually share the knowledge they already had, because there is so much surely that can be done with information already out there. They're just not sharing. And it's how can we do that? How can we get them to share that?
Right. Because I think, as you pointed out, Eva, there's all the data from the animal trials that were found to benefit the animals, but not the humans. And so, so there's, assuming they were done well enough to have useful results, which is possibly not the case. But yes, there's potentially a huge amount of useful information there for animals, isn't there?
Absolutely.
And animal treatments. But
it would be interesting to see how many of them were then translated into any sort of veterinary medicine, or whether actually, because those two arms are also kept very separate, they carried on doing animal experiments for veterinary medicines, which is also done in the same way that it is for humans. But if there was actually the initial interaction, perhaps those could have been avoided altogether if they were found to be helpful in the animals. So it's and I think just to sort of build up on what Joe said, because we sort of about trying to get people to work more closely together. I think that's the most important thing. And I think also, as you covered in your presentation, the thing is obviously with the experimental animals is they live in such hot environments. The disease is induced, and disease doesn't happen that way naturally. Whereas the companion animals that live with us, they pretty much reflect our own lifestyles often. You have the people
Same
who
diet.
have the type 2 diabetes, who may have the dog with the diabetes. You may have the obese person who has the obese cat or the obese dog. So I think it's more about perhaps also trying to understand more through the sentinel approach as to how we can learn about the diseases that are occurring naturally,
Yeah.
and how through working together we could actually try and help both sets of patients without having to harm anyone along the way. I think that's the emphasis.
Absolutely.
And I think is a sort of a listening to your presentation and also the conversation that we're having, because obviously often it is the money that talks. It does seem a very short-sighted approach through so much money being wasted over the last decades of not actually progressing and developing more treatments for diseases, just through the pure convenience of sticking to something that has always been done. Because it is, and it is good to see in many ways some of the pharma companies actually investing large amounts of funding into the organ-on-chips and the organized work, because from their point of view, financially, if they don't have to spend 12 years on getting a drug license and spending billions on it, then it would be beneficial for the patients as well as for the companies themselves.
Yeah, absolutely. I mean, you know, when you look at it logically, it all makes complete sense to abandon animal models from any way you look at it, economically, scientifically, ethically, you know, time-wise. But yes, it's because we're social creatures. We can't just look at the evidence and say, okay, well, that doesn't make sense. You know, when your lives and your livelihood depends upon it, it kind of gives it a different sort of angle, doesn't it? I mean, we have to, I mean, as a background in the social sciences, you've always got to be aware of the social structures that prevent us from being logical in a way. And I think that's what we're struggling with at the moment, really.
Yes, but we are all very hopeful for the future.
Yes.
So I'm going to just, because we've reached our time, thank you very much. It has been really, really good presentation. Really enjoyed it. Learned loads, have loads more to look up. So thank you very much, and thank you everybody who joined, and I hope the people who listen to the recording later will enjoy it as well. Well, have a lovely sunny afternoon and a lovely week.